The number of years a person has lived, known as chronological age, has long been the default measure of aging, but it often fails to capture the true condition of an individual's health. In contrast, biological age seeks to reflect the actual functional state of the body's cells, tissues, and systems. It is increasingly viewed as a more meaningful predictor of disease risk, cognitive decline, physical performance, and even mortality. While two people may both be 60 years old chronologically, one might exhibit the biological characteristics of someone a decade younger, while the other may show signs of premature aging. This disparity underscores the importance of biological age as a more personalized and actionable metric. Advancements in molecular biology and systems medicine have provided scientists with increasingly precise tools to estimate biological age. Among the most promising and widely used are epigenetic clocks, which assess patterns of DNA methylation, a chemical modification of DNA that influences gene activity without altering the sequence itself. As we age, these methylation patterns change in predictable ways, allowing researchers to construct algorithms that estimate an individual's biological age with remarkable accuracy. Pioneering models such as the Horvath and Hannum clocks have demonstrated strong correlations with chronological age, while newer iterations like GrimAge and PhenoAge have been developed to better predict health span, disease onset, and lifespan. Beyond DNA methylation, other biomarkers are being explored to enrich the assessment of biological age. These include telomere length, the protective caps at the ends of chromosomes that shorten with each cell division, inflammatory markers like C-reactive protein and interleukin-6, and metabolic indicators such as glucose tolerance and insulin sensitivity. Composite biological age scores often integrate these diverse signals to offer a multidimensional view of physiological aging. What makes biological age especially compelling is that, unlike chronological age, it can change direction. Numerous studies have shown that lifestyle interventions can slow, halt, or even reverse certain aspects of biological aging. Caloric restriction, plant-based and anti-inflammatory diets, intermittent fasting, regular physical activity, stress reduction practices, and quality sleep all have measurable effects on epigenetic age and other biological aging markers. For example, recent clinical trials have shown that structured interventions involving diet, supplements, and stress management can reduce DNA methylation age within just 8 weeks, suggesting that aging at the molecular level is far more dynamic than once believed. Exercise, in particular, has emerged as a powerful modulator of biological age. Both aerobic and resistance training influence muscle function, cardiovascular health, and systemic inflammation in ways that correspond with slower epigenetic aging. Similarly, mindfulness practices and psychological resilience have been associated with more youthful biological profiles, possibly due to their effects on cortisol regulation and immune function. Even moderate improvements in lifestyle can result in meaningful shifts, indicating that the trajectory of aging is not fixed but highly responsive to behavioral choices. Importantly, the implications of biological age go beyond personal optimization. In clinical settings, it can serve as an early warning system for identifying individuals at risk for age-related diseases long before symptoms appear. In public health, it opens new avenues for prevention strategies that are tailored to an individual's biological state rather than their calendar years. And in longevity research, it provides a quantifiable measure to evaluate the effectiveness of emerging anti-aging interventions, from senolytics to regenerative medicine. The growing accessibility of commercial biological age testing, many of which use epigenetic data, has sparked interest among consumers, though not all tests are equally validated. Still, the core insight remains. Aging is not solely a matter of time passing, but of how our biology responds to life's cumulative exposures and choices. The concept of biological age represents a paradigm shift in how we understand and approach aging. It offers a more precise, personalized, and optimistic view of human aging, one in which we are not passive recipients of time, but active participants in shaping how well and how long we live. By tracking and improving biological age, we are learning not just how to count more years, but how to make those years truly count.